Because proteins are made up of so many amino acids. So many amino acids. How many amino acids are involved in the synthesis of protein? How many amino acids are involved?
Sir, homopolymer.
20.
How many
20.
amino acids?
20.
20 amino acids are involved. Okay, so the structure of protein. Sure. So what you will have to know about that? That proteins are heteropolymer, contain string of amino acid. Okay, that is that proteins are what are proteins are? Proteins are heteropolymer. Heteropolymer containing a chain of amino acids or a string of amino acid, a string of amino acids. Okay, strings of amino acid. So proteins are heteropolymer containing a string of amino acid. In biology, there are four structures of protein. In biology, there are four structures of protein. What are these four structures? What are the four structures? The first structure is about that of primary one. That is primary structure. Okay, first structure is primary structure. It is called 1 degree structure. It is also called 1 degree structure. That is primary structure. The second one is that of that is very important one. That is, you have to know that is secondary structure. That is secondary structure. Secondary structure, that is 2 degree structure. 2 degree structure. The third one is that of tertiary structure. Tertiary structure. This is 3 degree structure. 3 degree structure. And the fourth one is that of that is quaternary structure. Quaternary structure, that is 4 degree structure. 4 degree structure. Okay. So today we'll have to know about that of structures of protein. That in biology there are 4 structures of protein. One is that of primary structure. Second one is that of secondary structure. The primary structure is 1 degree structure. The secondary structure is known as 2 degree structure. Tertiary structure is known as 3 degree structure, and the quaternary structure is known as 4 degree structure. So one by one we will have to know about that of different structure. First of all, we know about that of primary structure. Okay, what are the primary structure and what are the examples of primary structure, and that of the formation of the structure of that primary one we'll have to know now. So you have to know about that of primary structure. That is primary structure. It is very important. So first of all, I am just telling about that of primary structure. Okay, that you see that you know that is proteins are made up of different type of amino acids, and such amino acids that is binds to each other by peptide bonds. Okay, that is peptide bonds bind each amino acid and make a one polypeptide chain. Okay, one polypeptide chain. Suppose you see that you know that amino acid. This is the C, and here is hydrogen. Here we will have to know about that of double bond O. Here is H, and here you know that that is OH. Here is OH, and here is that of R variable. And here is that of amino group. Okay, that is amino group H and hydrogen. So here is that is amino acid. Okay, that is the structural formula of amino acid, in which you can have to know about that of this one is that of acetic acid. That is the acidic part. This one is that of amino group. This is the acidic group. That is carboxyl group. Carboxyl group. And this is the amino group. Carboxyl group is that of acidic in nature, and this amino group is basic in nature. Basic in nature, and this is acidic in nature. And this R is variable. but hydrogen is constant. Here hydrogen is constant, but R is variable. If R will change its place by H, it will be glycines, or it will have to different amino acid if the R will be changed by any other group. Okay, so you have already know about that how I have already shown you all the 20 amino acid in that previous classes, so that no need of discussion here. So you know that how two amino acids join together. Here you see that, that means here is a one amino acid and another amino acid will have to join here. Another amino acid will have to make, here is a hydrogen, here is that nitrogen, hydrogen, that is carbon, that is hydrogen, that is R1, this is R1, this is R2. And here is that C double bond OH. Okay, you see that. How can, I have already told you that in the bond formation that water is taken out, that means water will be taken out. That means here you see that here OH, that means this amino acid will donate OH. Another amino acid will donate hydrogen. That means water. So it becomes water H2O. So these will two go out and it will make a peptide bond. So here a peptide bond will form. Okay, a peptide bond will form here. Here will be a peptide bond. This is a peptide bond. Okay, peptide bond. That is, this bond is based on that of dehydration. All the bonds, peptide bond, disulfide bond, or that of any other bond, glycoside bond, I had already told you that. That means it is based on that of dehydration. So that whatever, that means this amino group, this amino acid will donate OH, that amino acid donated what? That is H. So OH, H, that means combine together, make water, so that water will go out and then a bond will form here. Okay, that bond will combines to that of that means this amino group to this amino acid to that of another amino acid. Like this, here also the same thing, there will be another bond. Okay, that is also a peptide bond. That is a peptide bond. So what remains? That is nitrogen, hydrogen, carbon, hydrogen. Here the R3, here that C double bond O. So another peptide bond you will have to find here. Like this. This is a linear chain. This is a straight chain. Okay, this is a straight chain. This is a linear chain. Okay, linear chain. That means so many amino acids will be involved in the synthesis of protein. It will make a linear chain. It is a straight chain. Okay, straight chain. You see that? That means how that one amino acid combined with another amino acid, and in such combination there is a formation of peptide bond. Like this, the second amino acid combined with that of second amino acid combined with that of third amino acid and also make the form of peptide bond. Like such process will move on. Like this, the process will move on by the formation of the peptide bond. So primary structure is completely based on that of a peptide bond, and it is a polypeptide chain. So primary structure is a polypeptide chain. Polypeptide chain, and it is a linear one. It is a straight one. Always primary structure is a straight one, is a linear one, and it is based on one polypeptide chain. One polypeptide chain. Why it is called polypeptide chain? Because so many peptide bonds are involved in this process. Here is a one peptide bond. Here is another one. Here is a third one. It is also a peptide bond. So there are so many peptide bonds involved in this process of that of a structure of protein. So this one is called one polypeptide chain. Okay, that is polypeptide chain, and this is a linear chain. Always remember that, and it is a 1 degree structure. It is a 1 degree structure. Okay, and the example of that of a primary structure, if you have to cite the example of that one, we'll have to know about that of aquaporin. Okay, one is that of aquaporin. I had already told about that of aquaporin in that plasma membrane. You might have remembered that. Okay, that aquaporin, how it is, that means helps in that of transfer of water from that of outside to that of plasma that is cytoplasm. Okay, so that aquaporin, it is a aquaporin. One example is that of aquaporin. Another one is that of a premature protein. That means protein, not mature protein. That is premature protein. Premature protein. Premature protein are the primary proteins. Those proteins are synthesized in ribosomes. They are premature proteins. So premature proteins and aquaporins are the example of that of a primary structure. Okay, primary structure. And this part is called, this part is called N-terminal. This part is called N-terminal. N-terminal. That is the first part. This one is that of a last part. That is N-terminal. This one is called C-terminal. This one is called C-terminal. Okay, that means the bond starts from that of N-terminal. This is called the first part, and the bond, that is this chain, this structure, ends at that of C-terminal. This called that ends part. That is the terminal part. This is the N-terminal, and this one is the C-terminal. N-terminal, this is the first part. This is the first part. C-terminal, that is the end part. End part. So that is N-terminal. This one is the C-terminal. And this is that of a structure. This is the primary structure. So many amino acids combined together form a primary structure. Okay, primary structure, and it is always a linear one, straight one. That is 1 degree structure. And examples, and it is a one polypeptide chain. It is a one polypeptide chain. So many peptide bonds join together form a one polypeptide chain. And examples of that is aquaporin. Another one is premature protein. Okay, premature protein. These are the example of that of a primary structure. Okay, that is primary structure. Then we will have to know about that of a secondary structure of a protein. This is the primary structure. Always remember that primary structure is 1 degree structure. Primary structure is a linear or straight structure. It is a straight line. It's like a straight line or linear one. And this is a one polypeptide chain because so many peptide bonds are involved in the process of that of formation of a primary structure. So many peptide bonds are involved. So it is called a one polypeptide chain. Examples to that of aquaporin and the premature protein. This is all about that of primary structure. You only need examination question on that which type of structure? Linear one. That is a linear one. That is 1 degree structure. Example, aquaporin, premature protein. This much. And the first part is called the first, that is beginning. The first part, the beginning part is called N-terminal. The end part is called that of a C-terminal. N-terminal, you can have to ask doubt also persist in your mind why it is called N part, because it's the amino group. This is that side of amino group, and this is that of a carboxyl group. Okay, carboxyl group. So it is that of N part, N-terminal one. This is the C-terminal one. This is the carboxyl group, and that amino group it has taken as N. Okay, amino group. This is the carboxyl group. That means starts with amino groups, ends with carboxyl group. So N-terminal, C-terminal. This is the first part. This is the terminal end part. This is about that of a primary structure of a protein. Okay. Now come to that of a secondary structure of a protein. Now see that secondary structure of a protein. Okay. Secondary structure of protein, that is secondary structure of protein. Secondary structure of protein, that is 2 degree structure. 2 degree structure I have already told you, you might have remembered. Okay, that is secondary structure of protein or 2 degree structure. The secondary structure of protein is of 3 types. One is that of alpha helix. One is that of alpha helix. Okay. Another one is that of beta sheet helix. Another one is that of beta sheet. Okay. And the third one is that of alpha collagen, that is collagen sheet. That is collagen. Okay, collagen sheet. So that we'll have to know about that of alpha helix. Okay, alpha helix. Alpha helix, and that in alpha helix you'll have to know that. You see that in that primary one, it is just straight, the straight one. It is a linear one. If you'll have to, okay, if you'll have to do like this, okay, do like this. That means we'll have to do like this. That means you have to turn it like this process. So it is called alpha helix. It is called alpha helix. Some proteins looks like this. This is the structure of some protein. They are alpha helix. And this one also called that of, this is the N terminal. This is the N terminal. And this is also C terminal. Okay. And this one joined by that of hydrogen bond. You see that there are some, that is intra and inter. They are joined by that of a hydrogen bond. Okay, hydrogen bond. Hydrogen bond joins to that of one amino acid to that of another amino acid. So this one is that of alpha helix. Another one is that of a beta plated sheet. That is, you see that one is like this. Okay, another one is like this. So that this one is joined by, this one is joined by, and this one also joined, this one is also joined, like this process. So this joining part, okay, this joining part is called hydrogen bond. This is also hydrogen bond. Okay, that means you know that in primary structure, peptide bond, that is responsibility of peptide bonds joining the amino acid. Here is that of one is that in alpha helix. You see that alpha helix, that is twist and turn like this, twist and turn like this. You will have to do it. This is called that of alpha helix. Alpha helix. Alpha helix. That first part is N terminal, second one is C terminal. This one also that N terminal. This one also that of C terminal. This one also that of N terminal. This one also C terminal. Okay, and this one joined by that of hydrogen bond. Hydrogen bond joined together. So that of beta plate, it is a plate like, okay, it is a beta plated sheet. Okay, that is, it is a beta plated sheet. And you see that it is a sheet like. Okay, it is a sheet like. That means you will have to see that it is a sheet like structure. It is a helical structure. It is a helical structure, but it is a sheet-like structure. It is plate-like structure. You should have to share that it is a plate-like structure. Okay, plate-like structure. And that alpha helix, basically alpha helix are found in that of keratin in hair. Where it is found? It is keratin protein. Keratin protein. That protein is found in hair, in your nail. Okay, so that this is keratin protein. So alpha helix example, only example come in the examination. Alpha helix secondary structure consists of three types: alpha helix, beta plated, and collagen sheet. And in that, in that you have to find out that this alpha helix example is keratin protein. Keratin protein, and it is a helical structure. And two ends of the structure is joined by that of a hydrogen bond. Similarly, beta plated, it is a plate-like structure. So beta plated sheets, and this also one end joined by another end also by that of a hydrogen bond. Hydrogen bond also joined together. So this is called beta plated sheet. And this type of generally found in that of a fibroin. That is where it is, that is fibroin. Fibroin, that is a silk protein. It is a silk protein. Okay, it is found in silk protein. And a lysozyme also. That is lysozyme. In lysozyme also, that is lysozyme protein also. That is beta plated sheet, like that of beta plated sheet. So only example is important, students, here, and nothing. Okay, nothing is important here. Only that secondary structure alpha helix. Alpha helix example is keratin protein. This much you will have to know. And joined by that of a hydrogen bond. Similarly, another secondary structure, another type of secondary structure is the beta pleated sheet. That beta pleated sheet is joined by that of hydrogen bond. It forms a plate-like structure and a fibroin protein. Example is fibroin protein. Fibroin protein is a type of silk protein. Another one is that of lysozymes. Lysozyme, that is in human, that means you know that in our buccal cavity, such type of that is proteins are found as enzymes. Okay, proteinaceous enzymes and such found are that of lysozymes. Another one is collagen helix sheet. That is collagen helix sheet. Okay, that is collagen. You see that here what you are doing, that is one turn. Here you are doing one turn. But here in collagen helix, you will have to do that of 3 turns. You see that. That means you start with one turn. One turn. Another turn also you do like this. And in which, in middle, you will have to do. In middle also you have to, that is, we have to do, that is, we have to do also, you have to do also another round. Another round here also, and that one not doing well. Okay, so this one. So this one is called that of that is 3 helices. 3 helices joined together. 3 helices. Helices joined together. Joined together. Okay, together. That you see that this is first helices. This one is the first one. This one is the second one. This one is the third one. This is called 3 helices joined together. That is collagen helix. And this is basically found in collagen protein. Name is given, so it is collagen protein. Collagen protein. So generally question asked, that means you know about that. In secondary structure, one example is that of keratin protein. Another one is that fibroin lysozyme. Another one is collagen protein. So keratin protein is alpha helix type of secondary structure. Beta pleated protein, beta pleated sheet, that is this structure. Example is that of fibroin, that is a silk protein. Another one is lysozyme. Then that of collagen helix. Collagen helix is that of collagen protein, and it is 3 helices. Here is a 1 helix joined together. Here are 3 helices joined together. You have to do like this. Another one also you do like this, the middle one, and they are perpendicular to each other. They must be perpendicular to each other. Perpendicular to each other. To each other. They are perpendicular. You see that. Here you are looking there, they are perpendicular. This one is perpendicular, this one is perpendicular, this one. So perpendicular, all are perpendicular. That means perpendicular to each other. And they are also joined by that of hydrogen bond. Okay, so that they are perpendicular to each other. That is basic thing is that in collagen helix, there are 3 helices joined together, but they are perpendicular to each other. Example of that of collagen helix from name itself, that is collagen protein. Okay, so in secondary structure, it is basically a 2-degree structure, and it is of 3 types. One is that of alpha helix, second one is beta pleated sheet, third one is collagen helix. In that of alpha helix, it is just a one helix that is twist and turn. Okay, this one is a twist and turn, and it is joined by that of a hydrogen bond. Another and example is that of keratin protein. And the second one is that of a beta pleated sheets, and they are plate-like structure joined together by hydrogen bond, and the fibroin, that is silk protein, and lysozyme, that is fibroin is silk protein, and lysozyme are example of beta pleated sheet. Examples are very important here. And a collagen helix, 3 helices joined together perpendicular to each other. This one helix, two, and middle one is 3. Example is collagen protein. So in secondary structure, that means in which proteins that collagen structure are found, you forget that of alpha helix, beta pleated sheet, or that of that is collagen helix. You forget that one. But example you might have remembered. The first one is that of alpha helix. Example is that of example of alpha helix is, students?
So silk. Silk protein.
Alpha helix example.
The keratin protein.
Keratin protein.
Keratin protein.
You have to tell loudly why you are not eating.
sir आपण सुनलेच ना.
Or that Makar Sankranti, that means you are observing that fasting so that you are not telling. Or in beta pleated seed, you cite the example, 2 example you cite.
Fibrin
Silk protein,
protein and
lysozyme.
Silk protein is otherwise known as
Fibrin protein.
fibrin, very good, fibrin, very good. And a lysozyme and a collagen helix. In collagen helix, how many helices join together?
3 helix.
3 helices, and they are dashed to each other. They are dashed to each other.
Perpendicular to each other.
They are perpendicular to each other. Very good. And an example is that of collagen protein. Very good. Then come to that of a tertiary structure. Okay, that is tertiary structure. That is tertiary structure. You see that the tertiary structure will have to see that it is a 3-dimensional structure. That is tertiary structure. Tertiary structure, that is 3-degree structure. 3-degree structure, or it is also called 3-dimensional structure. 3-dimensional structure. 3-dimensional structure. That is tertiary structure. And biologically, it is very important structure. It is a stable structure. Okay, it is a stable one. And biologically, most that is important structure is tertiary structure. It is very important, students. You have to remember that. Okay, most stable and necessary for biological activities. It is that is more stable and mostly essential for biological activities. Okay, activities. It is a 3-dimensional structure, and this structure is more stable among the 4: primary, secondary, tertiary, quaternary. This one is more stable structure. So generally question will ask, which structure is more stable? You can easily answer that of tertiary structure. It is a 3-dimensional or 3-degree structure and most essential for biological activities. All the biological activities carried out by that of a tertiary structure. So enzymes are, that means example is that of enzymes. Okay, enzymes. That enzymes, you know that enzymes are also proteinaceous. Okay, that means proteinaceous. So that enzymes are the example of that of a tertiary structure. So tertiary structure example is enzymes, and you know that enzymes play key role in that of structure. That you have to remember, all the enzymes are made up of tertiary structure. Owing to that reason, this structure is more stable or more essential for that of biological activities. In generally, you know that primary structure is generally a linear one, a string one, a straight one. But in case of that of secondary structure, you make them of a helical one. Okay, double helical one. Okay, double helical one. You are also making that of a beta pleated sheet. But here you see that. In that of tertiary structure, if you have to move that primary string into a several in a different way, you see that if you have to make like this, this one will be a tertiary structure. Okay, this one will be a tertiary structure. That means this will move in a different position, and this will be look like that of a ball of wool, that is a woolen ball. Look like a woolen ball. Okay, looks like a woolen ball. That is important. You must have to. It looks like a woolen ball. It looks like a woolen ball. Woolen ball. Okay, ball, B-A-L-L, ball. woolen ball. So it is also that of that is N, and this one is C. This one is N terminal. This one also C terminal. This is looks like a woolen ball. You'll have to take it and make it like this. Okay, this one look like that of. You can turn it in different way. You can turn it several times. This is the primary one. This is the primary string. But you have to move like this way, not like that of secondary structure, like that of helical one. You have to move in different way. You can have to find like that of woolen ball. It looks like a woolen ball. This is a tertiary structure. Okay, tertiary structure look like a woolen ball, and it is more stable because enzymes represent that of tertiary structure. All enzymes represent that of tertiary structure. So biologically most essential, this structure biologically is more essential and it is more stable, and it is a three-dimensional one. It is a three-dimensional structure. Okay, that point you have to remember. And another one, it is made up of so many. In primary structure, they are made up of peptide bond. In that of secondary structure, that is made up of hydrogen bond. But here you can have to find several bonds. So many bonds are involved in such process. One is that of hydrogen bond. Okay, one is that of hydrogen bond. Another one is that of peptide bond. Hydrogen bond, peptide bond. Okay, peptide bond also involved in this process. Ionic bond. Okay, ionic bond, you can have to ionic bond also. That is hydrophobic bond. That is hydrophobic bond. Hydrophobic bond, that means it has for hydrogen, it has that is it has fear for that of hydrogen, so it is a hydrophobic bond, hydrogen bond, peptide bond, ionic bond, and that is also that of peptide disulfide bond. Another one is that of disulfide bond. Disulfide bond. Okay, so many bonds are involved. So many bonds are involved in that tertiary structure. You imagine that. That means it is not a simple one. It is some way complex one. As a complex one. So that means it is owing to that of complexity in nature. It is most essential for biological activities. And for it is complexity in nature, it is more stable. Okay, it is more stable, that you have to remember. Okay, so that tertiary structure, what you will have to remember, tertiary structure is 3D structure, or that of three-dimensional structure, 3D structure. It is also called as that of 3D structure, also called as 3D structure, three-dimensional structure, and more stable owing to that of different types of bonds. Okay, involved in the process of that is formation of that of proteins. So that of so many bonds are involved: hydrogen bond, peptide bond, ionic bond, hydrophobic bond, disulfide bond. So many bonds are involved, so more stable and the mostly essential for biological activities, because all enzymes are that of a tertiary structure. Okay, tertiary structure. Owing to that region, this structure is highly important, mostly important, that you have to. Okay, that is tertiary structure. And then we'll have to proceed to that of another example, students, that is myoglobin. Another example is that of myoglobin. Myoglobin. What is myoglobin? That is myoglobin basically found in that of muscles. And this is a oxygen pigment. That is myoglobin is that of oxygen carrying pigment found in muscle cells. That you have to remember. It is a protein. It is myoglobin is a protein. But what it carries? It carries oxygen. It carries oxygen. So it is oxygen carrying pigment in muscles. Oxygen carrying, as that of hemoglobin is the oxygen carrying pigment in blood. Similarly, myoglobin is oxygen carrying pigment in muscles. That is, question will generally ask, which one is that of oxygen carrying that is pigment in muscles? Answer will be myoglobin. Okay, so that you have to remember that point. Myoglobin is also example of that of a tertiary structure. Okay, myoglobin is otherwise known as oxygen carrying pigment in muscles. That is very important. You must have to remember. Then students will have to come to that of that is last one, that is quaternary structure. Okay. Then you might have remembered all these things: 3D structure or three-dimensional structure or 3D structure, more stable, most essential for biological activities because of that of enzymes. Example, enzymes, and it looks like that of woolen balls, and so many bonds are involved: hydrogen bond, peptide bond, ionic bond, hydrophobic bond, disulfide bonds. And that example, another example is that of myoglobin. Significance of myoglobin is that it is oxygen carrying pigment in muscles. You might have understood clearly, and 100% sure that you might have no doubt. If you have consistency in your mind, you are not thinking otherwise, if you are concentrating in that of lecture, you might have understood everything about that of tertiary structure. Then students come to that of quaternary structure. Quaternary structure, this is the last one, and this will be otherwise known as the dash degree structure. Quaternary structure is dash degree
4
structure.
degree structure.
4 degree structure.
4 degree structure.
4 degree structure. So now we'll have to know the last structure of protein, then we'll have to finish that of protein. Okay, then we'll have to proceed to that of nucleotides and nucleic acid. And that means one students that Mona Lisa, are you present here?
Yes, sir.
And we had that on WhatsApp you had sent that message that you have doubt in that of amino acids and that of lipids. Okay.
Yes, sir.
Try my best to that, and some students also, so many students also absent on that period, that in 2 periods what I had finished amino acids and that of lipids. I will try my best to complete it, and today also in that of evening sessions also I'll have to give you that of knowledge, and the students who are already present and those who are absent, they can have to attend and clear their doubt in that of amino acids and that of lipids. Okay, students, now come to that of tertiary, that is quaternary structure. Quaternary structure, that is 4-degree structure. Okay, 4-degree structure. And that quaternary structure, you can have to, the best example of that of quaternary structure, student, is that of hemoglobin. Okay, that is of hemoglobin. Hemoglobin, that HB. Okay, hemoglobin. That we will have to know about that of hemoglobin. You see that here is that, you know, that hemoglobin, that iron they supply to that of iron. Hemoglobin is very essential, and also carry that of that is iron it carries. So that if you have to draw it, that structure, okay, first of all you have to draw it. You draw it to that structure, and here you see that you have to join them. Okay, these are, you see that there is Fe2 plus, here is also Fe2 plus, here is also Fe2 plus, here is also Fe2 plus. Okay, and you will have to find here that, what you will have to find here that they are of that of 4 subunits. You see that here is a 1 subunit. Here is a, here you will have to find that another subunit. Here is another subunit. Here is also another subunit. Okay, and this subunit, this is alpha subunit. This one also alpha. This one is beta. This one is also beta. So they are consist of 2 subunits. 2 subunits, 2 alphas, 2 alphas and 2 betas. 2 alphas and 2 betas. So hemoglobin consists of that of 2 subunits. One is that of 2 alpha, another one is 2 beta. Okay, 2 beta. As they are made up of the 4 subunits, 2 subunits are that of alpha, so 2 alpha subunits, 2 beta subunits. So 2 alpha, 2 beta, that is hemoglobin. And hemoglobin, the best example of that of quaternary structure. Okay, quaternary structure. And as you know that the basis of that of element, that of iron. So that hemoglobin, that you know that if RBCs that is not adequate in our blood, then you are suffering from that of anemia, and that anemia is that of due to that of deficiency of that of iron. So iron deficiency, that is Fe2 plus. So Fe2 plus, this is the structure, quaternary structure, and this one that of that is subunits. These are the subunits. 1, 2, 3, 4. 4 subunits. 2 subunits, 2 alpha and 2 beta. Okay, another one, students, is insulin. You might have known that about insulin. Insulin also a protein. As hemoglobin is a protein, insulin also a protein. So insulin also 2 alpha and 2 beta. It has also 4 structure. It has 4 structure, 4 subunits. 4 subunit, quaternary structure, 4 degree, 4 ones. So 2 alpha, 2 beta. So insulin also 2 alpha, 2 beta. Hemoglobin also 2 alpha, 2 beta. This is that of a structure, quaternary structure. And in this structure, what you will have to find that this is a more than one polypeptide chain. Another point in quaternary structure, you have to remember that that means primary structure, secondary structure, tertiary structure are made up of only one polypeptide chain. Only one polypeptide chain make that a primary structure. You have to make it twist and turn, make that of secondary structure. That is also one polypeptide chain. In that of tertiary structure, only one polypeptide chain, but you have to make it round in different ways, so that it looks like that of what? That is a ball of woolen ball, like that of woolen ball. That is tertiary structure. But here, not one polypeptide chain. That is not a one polypeptide chain. More than one polypeptide chain are involved in that of quaternary structure. For that of primary structure, secondary structure, tertiary structure, only one polypeptide chain is essential. Only one polypeptide chain. That primary structure can be converted into that of secondary one. That primary also converted into that of tertiary one if you have to move in such a way, and you will have to make it is a woolen ball. But in case of that of quaternary structure, there are 2 polypeptide chains are required, not one. Okay, that you have to remember. More than one polypeptide chains are required for that of quaternary structure. Okay, that concept should be clear. That is very important concept. Okay, more than one. You must have to remember more than one polypeptide chain. What I am writing? Polypeptide chain is required. Okay, is required. So basically two.
Yes, sir.
More than one. That concept, all the first, second, third, primary, secondary, tertiary of a one polypeptide chain. But here, more than one polypeptide chain. That concept is very important. And made up of 4 subunits, that is 2 alpha, 2 beta. 2 alpha, 2 betas, 2 alpha, 2 betas. If you not count them, there are 4 subunits. Okay. And so that is quaternary structure that you will have to remember, and this is very important one. And basically that question will ask about example. Example is that of hemoglobin and that of insulin. Okay. If you might have remembered, students, I'm just asking you, will have to tell me that that example of that of primary structure. Example, which proteins? Example of that of primary structure.
Sir, aquaporate and premature protein.
Yes, very good. And that of secondary structure, speed little. Yes, aquaporine, Jyotiranjan, very good, brilliant.
Sir,
Aquaporine,
keratin
that is
protein,
the first one,
secondary.
primary one. I'm just telling you, secondary one. That is.
Sir, keratin
hydrogen.
protein.
Hydrogen, yes.
Keratin protein.
Yes, very good. Aditya, are you present?
Yes, sir.
Okay. And that Anamika, are you present? Okay. That means collagen. Yes, that is also collagen. And example of that of tertiary one. That is tertiary one.
Enzymes and myoglobin.
Yes,
Myoglobin.
enzymes. Very good. All enzymes. In a special, that means I will have to teach you. It will be taken the 2 classes for that of enzymes. Very important. all enzymes and myoglobin. Yes, Jyotiranjan. Then that of that last one of the quaternary.
Sir,
That is
hemoglobin.
Hemoglobin and insulin.
yes, very good.
Hemoglobin and insulin.
Insulin and that of
Hemoglobin.
hemoglobin. Okay, students. So in this way, we have completed that of protein. Okay, protein. Now come to that of nucleotides. Okay, that is nucleotide and nucleic acid. That is nucleotide and nucleic acid. Nucleic acid, nucleotide and nucleic acid. All the nucleic acids are made up of nucleotides. Example of nucleic acid, students, have you remembered the example of nucleic acids? The two nucleic acid. Give me name of two nucleic acid.
DNA and RNA. DNA and RNA.
So today we'll have to know about that of nucleic acid means DNA, RNA, and how such nucleic acid are made up of a nucleotide. Okay, nucleotide. So nucleotide are basis for that of preparation of that of nucleic acid. So nucleic acid are made up of a nucleotide. Okay, nucleic acids are made up of a nucleotide. Now we'll have to know about that of nucleotides are compounds of carbon, hydrogen. So you write on that nucleotides are nucleotides plurally. That means plural. You have to nucleotides. Nucleotides are compounds of hydrogen, oxygen, carbon, nitrogen, and sulfur. Okay, hydrogen, carbon, hydrogen, oxygen, nitrogen, sorry, and phosphorus. Okay, hydrogen, oxygen, carbon, nitrogen, and phosphorus. So what are nucleotides? Nucleotides are compounds of hydrogen, oxygen, carbon, nitrogen, and phosphorus. That point you have to remember. In nucleotide, you can have to get hydrogen, you can have to oxygen, then we can carbon, nitrogen, and phosphorus. Okay, and the nucleotides, that means consist of three groups. One is that of nucleotide consist of three groups. What are the three groups? You will have to know. One is that of a nitrogen base. Okay, that is nitrogen base. One is that of a nitrogen base. Another one is phosphoric acid. Second one is that of
Phosphoric acid.
Okay, phosphoric acid, nitrogen base, phosphoric acid. Then we'll have to know about that of pentose sugar. The third one is that of pentose sugar. Pentose sugar, you know that already. That is which type of? That is monosaccharides. Okay, monosaccharide. That means which type of carbohydrates involved in that of nucleotide, formation of nucleotide. These are all monosaccharides. All that is monosaccharides. Okay, that is pentose sugar. Pentose sugar, monosaccharides are involved in that of nucleotides. So you remember that nucleotides are compounds of hydrogen, oxygen, carbon, nitrogen, and phosphorus, and consist of three groups. What are these three groups? One is that of nitrogen base. Another one is that of phosphoric acid, H3PO4, phosphoric acid. Okay. Another one is pentose sugar. Which type of sugar? Pentose sugar. That means monosaccharides. Okay. That points you have to remember. Okay. So you tell me that nucleotides are that is compounds of dash, and dash.
Hydrogen, oxygen, carbon,
nitrogen, and phosphorus.
Very good. And they consist of three groups. Which are the three groups?
Nitrogen based, phosphoric acid, and
Pentose
sugar.
Good.
Pentose sugar.
Nitrogen based, phosphoric acid, and pentose
sugar.
sugar. Now we'll have to draw that of structural diagrams. Structural diagrams we'll have to draw about that of nucleotides. Okay, nucleotide. I'll have to give you immediately after the end of the class. I'll have to supply you that of notes. Okay, don't think that I have been that means clean it, but you might have to be got as soon as possible. Okay, you see that I'm just drawing about that. Okay, you also draw with me simultaneously. You see that here is O. That is pentose sugar. Okay, pentose sugar. So that you have to, that means you see that here we'll have to draw a monosaccharide, a pentose sugar. That is, here is that pentose sugar. Pentose sugar. Okay, pentose sugar. And here we'll have to find out, here also we'll have to find out. There are one is that we'll have to find out that nitrogen base. Okay, here we'll have to find out that nitrogen base. Here is that nitrogen base. Okay, that nitrogen base is otherwise known as that heterocyclic compound. It is also known as heterocyclic compound. Heterocyclic compound. And here we'll have to get that phosphoric acid. Here we'll have to get that phosphoric acid. Okay, that phosphoric acid. Here is that phosphoric acid. And that you see that here is that pentose sugar. Pentose sugar attached to that of nitrogen base by that of which type of bond join it? This is called glycosidic bond. This is called glycosidic bond. Okay, this is called glycosidic bond. That means this bond join sugar to that. Here is carbon, here is a carbon, here is another carbon, here is another carbon, here is another carbon. Okay, so that another carbon also you'll have to get here CH2OH. Okay, I'll have to tell gradually. I'll have to. Don't be hurried or don't be panic. Okay, so that you see that is the nitrogen base. So the sugar joined with nitrogen base by means of glycosidic bond. Okay, glycosidic bond. And here is that sugar to be attached with that of phosphoric acid. Shortly I have written P. That means you consider it that it is phosphoric group. Okay, that is phosphorus. Phosphorus. So that H3PO4 instead of I have written the P, and this is joined by that of phosphoester bond. By that of phosphoester bond. Phosphoester bond. Okay, phosphoester bond. I have tell you that that means after that nucleotide nucleic acids portions finish, I'll have to the formation of different I have already told about that of glycosidic bond, and I'll have to tell about discussion about phosphoester bond, that is ester bond. Okay, hydrogen bond. I'll have to elaborate it again because there is a special portion in this chapter, and I'll have to tell you. Don't be. That means I will tell whenever is possible. Then you see that is pentose sugar. This pentose sugar joined with nitrogen base by glycosidic bond. Again, that pentose sugar joined with that of phosphoric acid by means of phosphoester bond. Okay, phosphoester bond. Again, you see that, and that means this is one, or this is what? That means this is a nucleotides. If you have to make another nucleotide, you see that you are making the another nucleotide. Okay, nucleotide. You have to make another nucleotide. This is also nitrogen base. This is also phosphorus. Okay, phosphorus. And you see that this is a 1, this is 2, this is 3. Okay, this is 3, and this is 4, and this one is that of 5 carbon. Here is that of CH2OH 5 carbon. Okay, so what you see that here what you will have to join. That means it is another nucleotide. This is a one nucleotide. This is another nucleotide, and one nucleotide joined by another nucleotide. Okay, one nucleotide to be joined with another nucleotide by phosphodiester bond. Okay, by means of phosphodiester bond. So this is, so you have to these three to be joined with that of P. That is called phosphodiester bond. This one is called phosphodiester bond. Diester. Because again also, if you have to draw another one, this one also phosphodiester bond. So each So each nucleotide, one nucleotide joins with another nucleotide by which bond? By phosphodiester bond. Okay, by phosphodiester bond. So this is that you see that this is a nucleotide. This is one nucleotide. Another nucleotide joins with that because nucleic acid are made up of nucleotides. So nucleotide, one nucleotide join with another nucleotide by means of a phosphodiester bond. This bond join one that is nucleotide to another nucleotide. So chain will continue. This chain will continue, and you see that this one is that of a carbon 5. And if on the above there are no other nucleotide, this one will be what? This one will be the 5' end. Okay, that is 5' end. You see that whenever you are drawing, that means you are drawing that of what? You are drawing that of a DNA. In that of a DNA, you are seeing that this is 5' end, this is 3' end, this is the 3' end, this is that of 5' end. You see that. Suppose there are no nucleotide on that, so that it will start with that of 5' end. And whenever you see that this is the 3' end, that is 3 carbon, carbon 1, carbon 2, carbon 3, carbon 4. Here is that of carbon 5. Suppose here is a nucleotide, here is a nucleotide. So, but on that below there is no nucleotide. There is no nucleotide. So there is a only that is a blank, that is carbon chain, that is carbon chain that is not attached to anyone. So this one will be the 3' one. And on the opposite one, if you will have to make it, if you will have to make it in that of opposite, okay, in that of opposite, you will have to make it. This one and another one I have also made. Another one also I also made like this. Another, you see that is a reverse one. This is a reverse one. This is also nucleotide. You see that in this nucleotide also, here is that of nitrogen, here is that of nitrogen, here you see that this is the nitrogen base. If you have to make it reverse, one will be the nitrogen. This one also that of nitrogen, and this nitrogen will have to combine with this nitrogen because they are parallel. Because they are parallel to each other, so this one will combine with this one by that of a hydrogen bond. Okay, and this nitrogen, suppose if I have to draw here, I am just drawing here. You see that I am drawing here. I am drawing here. Suppose this one is a nitrogen base. This one is a nitrogen base, and this nitrogen base is joined by another nitrogen base. By which bond? That is hydrogen bond. Okay, hydrogen bond join one nitrogen group to that of another. So that is, you will have to know that that is adenine, guanine, cytosine, okay, uracil. That I will have to tell because time is now on, and it is I will not hurriedly I will not tell because it is important one that you must have to understand. So you have to remember only this one, that nucleotides are compounds of carbon, hydrogen, oxygen, and then nitrogen and phosphorus, that you will have to remember. Carbon, hydrogen, oxygen, nitrogen and phosphorus, that you have to remember. And nucleotides are made up of 3 groups, that is pentose sugar, that is that of nitrogen base, and that of phosphoric acid. And if you have to draw a nucleotides, then this one is the oxygens, and this one is the first carbon. From the first carbon, nitrogen base starts, and this joined by the glycosidic bond. And this is the second carbon, this is the third carbon, this is the fourth carbon. This is the fourth carbon, and this carbon, the one side is by that of CH2OH, so this is C. This is the fifth carbon, and this one, this fourth carbon is joined by that of phosphoric acid, by that of phosphoester bond. And each nucleotide, if in between 2 nucleotide there is a one bond, that bond is called phosphodiester bond. Okay, phosphodiester bond joins that third carbon to that of a phosphoric group. That is phosphoester bond. You know that phosphoester bond. So that this phosphoric acid P is joined to that of a third carbon, not fourth or that of second carbon. So this bond is called phosphodiester bond. phosphodiester bond, that you have to remember. Okay, and that this much you remember, and in that evening session that you will have to complete that of nucleotides, nucleic acid. After I will have to make your doubt clear about amino acids and that of and another one is lipids. Okay, so now I am just asking you questions, and you are now ready, and you just have to write down the answer which one is the correct one. Okay, now you ready for that questions. So that is a surprise test. Okay, that is a surprise test. I'll have to only ask the 2 questions, and in the evening sessions I'll have to ask you 5 questions. In evening session I'll have to ask you 5 questions. Now I'm asking you 2 questions because you will have to go for lunch. Okay, I have already taken my lunch, and so that you should be hurriedly go for lunch. So you see that first question, students, you are now ready. You don't have to write the answer. Whenever I look that up screen, you have to write down the answer. Okay, don't be hurried, and don't whenever I'm writing, don't see that is show me the answer because I can see it, and so that there will be a controversy. So the first question is that which of the following is a non-reducing sugar? First question, which of the following? Which of the following is a non-reducing sugar? Okay, I'm just giving you option. I have not given options, so don't you write option A. I'm giving you options. First one, you write down that of lactose, B for maltose, C for sucrose, D for galactose. Yes, you just show me your answer. Yes, Tushar Ranjan, brilliant answer. Brilliant answer, Tushar Ranjan. Scored the good marks today. Yes, Anamika also. That answer is she, sucrose. Sucrose is a non-reducing sugar. All the students should have to show. Only 2 students have shown, other students remaining silent. What? That
Sir, I have
nonsense going
sent the
on.
answer.
Yes. So
Yes,
that
sir, I have
Vishwajit,
also
yes, don't tell the answer because I am not God
written
that
in
I
the
can
chat, sir.
listen to you all. Okay,
Sir, I've
I am a
written in the
human
chat.
being, I'm Homo sapien. Okay, so you just have to write down that yes, that Sandhi Parija, so let Sandhi
Sir, I
Parija.
have sent
Okay,
earlier.
you have taken so many times. Okay, so that the answer is sucrose. Today that Meher has scored that of 25 marks. Then the last questions of today, afternoon session, there will be 5 questions. Okay, so you be ready. Battery is 15% now. Okay, so that I'll have to charge the mobile. So just the last questions I'm writing. That select the incorrect pair. Second question, select the incorrect pair. Select the incorrect pair. Incorrect pair. Be ready. Don't tell the answer. Okay. Fructose. Fructose. Fruit sugar. Okay. Then B. That is ribose. Ribose. ATP. Okay, I'm just writing. You have to write with me also. Lactose. Lactose. Milk sugar. Okay, and that you glucose. Glucose. That is brain sugar. Now you answer. Now you answer. Yes. Yes, brilliant, Shanbhe. Adhyasa Dubey, little late. Tushar Ranjan, little late. Okay, that right. All answers are correct. Okay, so that Shanbhe and that today's that's highest score. That means Shanbhe fastest finger pass. First that Tushar Ranjan. Tushar Ranjan scored 25, and that of Shanbhe also scored that of 25. Thanks you, Tushar Ranjan and Shanbhe. That means
Thank
for
you, sir.
participating. All students should have to participate. Don't mind that. Okay, don't mind that because this is a type of competition. Okay, so Biswajit Mahalik, you have glucose told, sir. What told? I have not hearing to you. Okay, that Biswajit, you write down. You make practice. If you have to kaun banega crorepati, if you have to practice, you should have to practice here little bit. Okay, thank you, students. Joining, you go for lunch. Okay,
Thank you, sir.
thank you, students.
Thank you, sir.
Thank you,
Thank
sir.
Thank you, sir.
Nucleic acid, then you will have to clear your doubt of that of amino acids and lipid. Thank you, students. Thank you.
Yes, sir. Thank you, sir. Very much.
What nonsense. That's not happening.